All right, I want to welcome everybody to the Parrot Club's September 2024 meeting. Today we have Dr. George Olah of Australian National University talking to us about using DNA to stop the illegal parrot trade. Dr. Olah is a scientist and conservation geneticist, mainly working on parrots and other birds. He received his Master of Science degree in zoology at the University of Veterinary Medicine in Hungary and his PhD in conservation biology from Australian National University. He has participated in many field-based research projects on parrots in Central and South America. He worked for the Tambopata Macaw Project in the Peruvian Amazon for several years and has been a lodge manager for one of the largest Peruvian ecotourist companies. He co-founded Wildlife Messengers, a nonprofit organization creating scientific and educational film projects based on their importance for conservation and whether a resultant film can actually contribute to decision-making and protection. He is the principal investigator and conservation geneticist for this genetics project at ANU. And you can visit stopparrottrade.org to learn more. With that, I will turn it over to Dr. Olah and thank him very much for joining us today.
Thank you. Thanks, Amy, for the introduction, and welcome everyone who are joining us online tonight, but this afternoon, I guess, in the US time. So yeah, I'm just trying to share my screen and see if this works. All right, I think it's all good. So yeah, as Amy mentioned, today I'm going to talk a little bit about an interesting topic or different topics that merge together in this research project. I named this talk the CSI of Parrots, combining forensics and conservation to study and stop the illegal wildlife trade. You all might wonder, how these different things fit together, like criminal science and parrots, and also like genetics and wildlife trade. So I will try to explain all that in this talk, and also like if you have any questions during the presentation, feel free to raise your hand or just feel free to interrupt, and I can answer your questions on the spot, or can just wait until the end and we can discuss, or if you have more interested to more details in some parts of the of this project or the research, we can also talk. So yeah, feel free to ask anytime. So in this talk, I try to set up the structure as a beginning, talking a little bit about parrots that I'm sure you're all familiar with, so I don't need to spend much time on that, but just a little bit about the background and the status in the wild. And then I would like to go into the wildlife trade, and then how genetics can actually help this illegal activity. And then also the importance of outreach and communication in trying to stop of the illegal wildlife trade. So on the screen, you can see lots of macaws. This has been taken in the Peruvian Amazon in Tambopata. So of course, macaws belong to parrots. Many people in general who don't who don't keep parrots or not involved in parrots, as you probably all are. But the general audience often don't know, exactly what a parrot, like the description of parrot, fit into, like. And that's why I often show, for example, these pictures that, of course, macaws are parrots, but also cockatoos. These are goffins. We took this picture in Indonesia, in the Tanimbar Island. And these are goffin cockatoos. So, of course, cockatoos are often not as colorful as, for example, South American macaws and other parrots and parakeets. They mainly white or black, like the black cockatoos, or the Australian black cockatoos are quite interesting colors, or pink. But they don't have as many colors as like macaws. Of course, if you talk about colors, you can also think about lorikeets. So, for example, this rainbow lorikeet, they also belong to parrots, and as the order of birds. They also are very, very rich in species because they adapted to feed on nectar. So that's the main source of food, and this made like a huge diversification of species. So they are also parrots. Of course, in Africa, we also have some parrots. They, for example, the very well-known African gray parrot that are also quite, famous as pets and very well known about their intelligence. But that's also true in general about parrots. Like parrots have, like a very high brain to body ratio. And if we go to a very interesting place on Earth, that's like New Zealand, you can find the Kias there. If you look into the eyes, you can already tell that how intelligent and often very badly behaved parrots they are. And of course, they also belong to the parrots. Actually, they are one of the very ancient branch of parrots. And if we look at the taxonomy, the phylogeny of parrots, you can see that all these strange and interesting parrots, like the night parrots, the kias, they all belong to a very early split from the rest of the parrot order, which is we call in taxonomy it's as order as the Psittaciformes. And then we also have, of course, all the cockatoos and the cockatiels, and lots of species in Australia, of course, mainly because parrots and this whole order originated in the Gondwana in the southern supercontinent in the past. And then they radiated to the different other continents. So lots of radiation and spreading the species to Southeast Asia, all the islands, if you think about like Indonesia and like Papua New Guinea. And then, of course, Africa as well. Africa is not that rich of species as like other continents, but we have quite a few parrots there as well. And of course, South America has a big, big diversity of parrots as well. And you can see the colorful macaws and Amazon parrots, all that are also very well known in the pet trade as well. We're not sure exactly when parrots, the last common ancestors, started or originated from, but probably around 60 to 50 million years ago, so quite a long time ago. And we have an amazing diversity still of these species. If we count them today, we have around 400 species, plus or minus, depending which taxonomy you are following, because recently they also changed around a few species, some subspecies into species level, and also shuffled them around among genus and among genera. But if you look at the map, of the world, you can see like the darker green represents more species overlapping, so more higher species diversity of parrots. So you can see like South America is very rich in parrots, and also Australia and New Guinea. But of course, Southeast Asia and then Africa also has some. And now we also have some parrots in Europe, of course, that are found in the cities. But as you see into the status of these birds, unfortunately, we showed that 1/3 of all the parrots that still exist today, are threatened. One species, the Spix's macaw, is extinct in the wild, although now they're being reintroduced to the wild. So hopefully this status will also disappear and they become only critically endangered. But quite a few species, unfortunately, already have gone extinct. And species that belong to critically endangered, or vulnerable categories of the IUCN and Red List, commonly call these species as threatened by extinction. And so the number of these species is about 1/3 of all parrots exist today. One of the main reasons why these species disappear from the wild is habitat degradation. Here, for example, I just have a screenshot from one of our latest studies that we published. in the global change biology shows like a prediction by the year 2050, have the different wood extraction logging from these habitats going to accelerate. And unfortunately, you can see like, well, all parrots live today and hopefully in the future as well. We expect an even higher rate of habitat clearing for wood extraction. And that, unfortunately, as we predicted in this model, is going to push even more species up to this endangered level. So they probably going to become more threatened than the species today. But if we look at all the species and all the threats available, and we did this study with BirdLife International, we look like what percentage of the species are threatened by what kind of threats. And if you look at this, the three highest threats that threatening most of the parrot species today are agriculture, also logging, it's number 2. So these all kind of habitat modification, I mean like industrial level agriculture and logging. And as I showed also previously, you can see here, for example, that human modified landscape are going to be not less but more as we progress in time. So by 2050, also, species are going to be more threatened by these threats. But these are very big and global problems that unfortunately we can't really resolve by with one organization or one group of people, because this involves like a huge international efforts and collaboration to try to do different kind of logging and different kind of agriculture that won't threaten these species so much. Parrots are especially threatened by logging because they live only in mainly most species lives in hollows in big trees. So they not making those nests like other birds. They actually occupying these hollows already available. And if we log those trees and forests, they don't only lose the habitat where they live in, they don't only lose the foods they feed on, but they also lose the place where they can nest and reproduce. And that's a huge issue for future generations. If they can't reproduce, unfortunately, they're going to disappear slowly from the wild. But if you look at this chart, you can see that there's also a big threat, which is hunting and trapping for parrots. And most of other species, if we talk about other birds or mammals in general, habitat loss and human modification of habitat degradation, that all influences most of the species on Earth. But hunting and trapping for the trade is not influencing all species equally. And that's why I want to go back to these slides. Previously I showed these are such charismatic birds. They're very intelligent, they're very colorful. We all love them, and there is a trade on them. It's not something new that just happened. It's been going on for centuries. And unfortunately, that's causing these most of these species also extinct in the wild. So just comparing to these images, in Indonesia, for example, we see lots of the trade of the cockatoos, lots of confiscation there also, like small lorries I'm sure you saw these images, like they stuffed in plastic bottles so they can fit more birds into a track or transport them easily. There's a huge problem also in South America, Mexico, and all the Latin American countries where they have like direct access to these species in the wild. But of course, also in Africa, there's like sometimes hundreds of African greys being still poached from the wild. For example, these images were taken in the Democratic Republic of the Congo. Even though there's like a low breeding facility in South Africa, there's still going on poaching in the wild, and so, unfortunately, this is still an ongoing issue. Of course. lots of restrictions have helped to reduce the trade. But just looking at the magnitude here, if we look at the international parrot trade, and this is a report by Traffic, and looking at as the, for example, the CITES reports, we can see that if we take into account all the live birds that's traded, in the past decade, we can see that parrots take almost like 90% of the number of these individual animals traded on the wild. So while other things like ivory and rhino horn, things like for traditional medicines, they mainly use that and smuggle that as a product, so that's much easier to hide and transport, especially if they already processed, and like pangolin scales and like that. But this is a big issue because most of the trade for parrots, it means it's a live trade, like they trading live birds because they want they're not trading them because they want to eat them or not using them for medicine, but they want to trade it as like live birds for the pet trade. And of course, this is the illegal part of it. There's also a legal part of the wildlife trade and the parrot trade. That's mainly alleviated by the facts that now we have a really good breeding facility across most of the most of the countries worldwide. But unfortunately, this is still an issue in the wild. So what we did a few years ago, we wanted to better understand, like there are certain hotspots internationally to see which which countries are still trading lots of parrots and what's kind of fueling this trade in those areas. So we joined with criminologists who use the different statistics to analyze different crimes, mainly human crimes, and different trades, mainly like drug trades or armed trafficking. And we applied these models to study the parrot trades. And as a model, we took Indonesia, because like Indonesia is a big hotspot for trading trading parrots illegally. And I try to show you a video of, that better explains our main finding of that study. So let me just stop this because I need to share the. I just need to share the audio as well. So let's see. Okay.
Wildlife trade and utilization, whether legal or illegal, are responsible for the potential emergence and spread of many zoonotic diseases that can cause novel human diseases, stressing the importance of regulating these activities. High global and domestic demand for parrots as pets and consequent removal from the wild for the trade have significantly contributed to their severe decline worldwide. 1/3 of the nearly 400 parrot species are threatened by extinction today.
The trade is vast. Not every parrot species is at equal risk of being traded. There's controversy concerning the role of demand and the opportunity-based factors driving the illicit wildlife trade. We use the criminological model CRAVE to analyze the factors associated with traded parrots in Indonesia, a country shown to have the highest priority for parrot conservation.
Southeast Asia is both a major hotspot for biodiversity and an epicenter for illegal wildlife trade worldwide. We quantify the relative importance of the components in the criminological model. For this, we use data of previously published studies and recent parrot confiscations covering a timescale of 2 decades in Indonesia. We showed 3 factors predicting a higher probability for a species ending up on the wildlife trade. First, when it was possible to legally export the species. Second, when they were more attractive, like colorful, large, and able to mimic suns. And third, when their range overlapped with higher human population density. This suggests that demand and opportunity-based factors together can partially explain the illegal wildlife trade in Indonesia.
The finding that demand-side factors are important suggests that people are targeting attractive species that are easier to sell in illicit markets, and that there is a cross-cultural preference for particular parrot species, especially ones that have been historically overexploited. Significant overlap between the domestic and international trade of certain species suggests the possibility that a high number of wild-caught birds in Indonesia are purposefully mislabeled as captive bred in order to be legally exported. The non-significant effect of the status of national protection of these species indicates that Indonesian market sellers are not fearful of regulations, so more efficient law enforcement is needed urgently.
Kegiatan konservasi dan penyadartahuan masyarakat sangat penting untuk dilakukan agar keberadaan populasi yang ada di alam tetap terjaga dan terhindar dari kepunahan.
Further strategies to reduce the trade of a studied species could take the form of nest protection, educational awareness campaigns for children and consumers, and using the power of media in science communication.
We have a program of CAP, Conservation Awareness and Pride Program, so make people in here proud, so they can also help us to protect the bird, especially cockatoo and parrot.
So you could see that it is very important to also understand what the root of the problem is, why people are trading these parrots, and that's what we try to answer with that study and find that, like education and information, spreading this information about this is illegal and what other alternatives there are for people who are doing these activities are very important, and I'm going to return that later at the end of my talk to this, because this is also a very interesting and important collaboration we have in Indonesia with the Indonesian Parrot Research. So at the outreach part, I'm going to talk about it a bit in more details, but that's not the only tool that we use to study the trade, and we use genetics specifically in my research that focusing on parrot genetics and studying parrots with genetics. It's been around for from the 1980s, and this is just a quick overview. And if you're interested, or maybe if you don't know much about genetics in general, I very highly recommend you to read. This is a very interesting paper we published in Diversity. It's open access, or I can send it to Amy, and Amy can send it to the others. It's like a whole overview of all the different studies that's been done on parrots in genetics, and how these different genetic studies have helped to inform our understanding about parrots. So this is like just a quick slide about, we've been using genetics, for example, from the 90s to molecular sex the parrots to tell, males and females apart. It's still actively used today. Just a drop of blood or just a feather sample is enough to tell whether it's a male or female. Then in the 2000s, the so-called Sanger sequencing allowed us to sequence the mitochondrial DNA that helped to better understand phylogenetics. And then new different genetic markers from the 2000s even let us to identify not just species, but even down to the level of individual identification. And of course, with the huge advance to the next generation sequencing technology, we could now sequence the whole mitochondrial genome, or even the whole nuclear genome of the species. And many of the species, this genome, and I'm talking, of course, about parrots here, are already sequenced and being sequenced now and uploaded to international databases as well. And then we can use different, next very advanced genomic techniques these days to look even deeper. And sequences is also getting cheaper and cheaper today. So that also contributing to, more sequence data and better understand it. And with the long read sequencing, for example, the Oxford nanopore technology now even lets us to take a sequencer to the field, because it's like the size of a USB these days, and you can even sequence that, in situ, and you can get, immediate information about those sequence data. So we can, for example, use that to identify a species or like a group of individuals, provenance at the site. And I just want to give you a few examples. Not all of these are, of course, like our research, but how genetics, for example, helped so far with the parrot trade. This case study happened in Australia. Someone detected, a person who was climbing up on a ladder to a tree and removing something from a nest, and this was in a protected area. So we reported it to the police. They took picture of the license plate of the car. So the police went out. They find a person. Turned out that this person was actually a parrot breeder and in a nearby town of this protected area. So what they also found that this tree where they took the picture of the person climbing the tree, there was a nest, and they found eggshells inside the nest. So they were not sure, what species are living in the nest, but they took these eggshells, you can see on the image here. They took those as samples, as you could see, for example, in a CSI movie, that they take the evidence, they put it in the Ziploc bag, they label the evidence, send it to the police lab. They also found, a very young chick of a black cockatoo, a red-tailed black cockatoo at the breeders, and he didn't have any of the, like adult of this species. So this was the only chick. So they also took, I think, a feather sample from the chick, and then also sent it to the lab. It turned out that they could match these two different DNA profiles, the eggshell and the bird, exactly, that it was a complete match, which means, using this genomic marker analysis that they also use for humans, for example, if they trying to identify, a criminal, that it was a complete match, and this actually led to criminal conviction of the person, a fine, and also he had to pay for the court cost. Of course, this is Australia, so they took it much more seriously, that this might not have happened in a country like Peru or somewhere else, where it's not the main the main issue of law enforcement. So this was a poaching case. There's also another case from Australia that involves smuggling of eggs. So this man was stopped at the border force in Australia at the international airport, and coming into Australia, I believe. And they found these kind of vest that has little pockets, and that in the little pockets he had eggs, small white eggs. So of course, from the eggs, the police could not identify what species they belong to. So they took DNA samples from the eggs as well, and sequenced the mitochondrial a mitochondrial gene that's been used for species identification, kind of a barcoding, and then they matched against an international database that's available online. And then they found out that most of these eggs belong to parrots. And of course, like CITES listed parrots, that you need, CITES permits to import or export these eggs. So again, the smuggler was prosecuted because there was a genetic evidence that they could use genetics to identify the eggs. And of course, they didn't have to wait for the eggs to hatch, etc., to finally identify the species, because that would be not so feasible, in a case like this. Interestingly, there was also a similar case in Brazil. I think in Rio de Janeiro, they also confiscated someone who's been trying to smuggle eggs on a plane. and they did a very similar mitochondrial DNA identification, and they find out that was about 57 parrot embryos in the confiscated eggs. So unfortunately, compared to Australia, these results were not used in investigation. They were just published in a scientific paper, as it was a very interesting case study. But as I said, being Brazil, these cases were not treated so rigorously like if it would have been like a human crime. But fortunately, these are just a few examples that these DNA evidences that we mainly use, and it's already been accepted for human criminology. It's being used for wildlife crimes as well, and especially for trade. It's very important. It's a very important tool. Another big area that DNA can help us. It's not only to identify the species we're dealing with, which is also, of course, it's like the number one that we need to know, but also once we identify the species, next, we want to know where they're from or where these individuals are coming from. And this is already my research that I've done in Peru. And I'm sure you remember the macaw project that we talked about earlier, that we collected, feather samples in the Peruvian Amazon from macaws. And also using genetics and extracting DNA from these feathers that we found all across the landscape around the clay licks. We identified like each feather we tagged like a unique barcode. We identified like this is individual A, this is individual B, etc. And then we find lots of matches in our database. For example, that we find 2 or 3 different feathers found in different locations, all belonging to the same individual, just like left at different places. So this is kind of like a forensic case because we identify individuals by their genotypes, just the DNA. But for research, we can also use this technique to identify different subpopulations or and see different how they move around. So that was our main case in the macaw project, was to better understand how the populations are connected and how they're moving around, which clay licks they visit. But of course, if these parrots were happen to show up in a confiscation, for example, in Lima, in Peru, and we try to match it against our database, then we would have a certain confidence to assign them one of the populations that we sampled, which is also quite an important information for law enforcement and also for rewilding, as I'm going to talk a bit in a bit later. Also, similar project in Australia on a major Mitchell's cockatoo. They sampled all across the Australian continent, and actually they find out that these guys from the east are very different from the west, and they actually belong to 2 different subspecies. And they also were able to draw a line where the 2 subspecies split. So again, they worked out like a genetic test that can be applied to any individuals, not just from the wild, but the ones that in confiscation or kept in a zoo. And then they can assign which region or territory they coming from. Another case that our research group, we were just talking about the difficult bird research group with Amy before. This is a research group that mainly focusing on very difficult birds, and difficult, I mean, it's really hard to study them, and also they're very rare and often quite endangered. So this is the case with the swift parrot, which is a critically endangered parrot in Australia. If one of the nomadic and migratory parrots there, the other being the orange-bellied parrot, which is also critically endangered. And they feed on nectars, and they need to find and follow the eucalypt trees because they not only feed on these trees, but they also nest into these hollows. And of course, these trees although they find all across this part of eastern Tasmania, they also flower different years in different regions. So they actually need to go and find these nests and these trees. We, again, our colleagues took DNA samples from these birds, and then we compared them. They all turned out to be one single meta-population, like, the whole species being a one population, just going to different parts. And so also our genetic evidence showed that they are heading towards extinction, just as the population model predicted, just based on demography data from the past. And also we showed, based on purely genetics, that they also change their mating system. There are more and more extra paternity detected in the nest. So, like chicks in the same nest are not full siblings, but often half siblings, or sometimes not fully related. And this also reduced the viability of the population. It's kind of like an extinction vortex that they're getting into. And what we were interested to know also using, purely genetic data, there's a thing called, you can calculate the effective population size, which you can use to predict the actual population, the number of individuals in the wild, the census population size. And if you look at this graph, you see, these are the estimates just based on observation, and it's coming from the past, that it's probably like between 1,000 or 2,000 individuals still left in the wild. And our genetic models actually showed that there are actually less than 300, or something like 300, 500 individuals, and these we double checked it with newer genetic tools, and that also confirmed that yes, there are probably less than 500 adult individuals left in the wild. So you can also use genetics to indirectly look at also to predict the number of individuals that's still surviving in the wild. And on the same line with this research, another interesting CSI and genetics combination came along when we looked at one of the, well, not us, but like our colleagues, but we all knew that that the sugar gliders, which is a nocturnal marsupial, are predating these nests in Tasmania. So they not only take the eggs, but often they kill the females that incubating the eggs, which is a huge problem for the survival of the species. But in Australia, because these are native marsupials, they all protected by law, so you can do anything about the eradication. But there was a hinge that they likely introduced to Tasmania, based on their distribution over time. You see that it started here, and by 2015, it's almost across the whole island of Tasmania. But there was no strong evidence. So a group of researchers did like a museum genetic project where they took samples from, the same species of this sugar glider across the whole range of their distribution, which, you can see on the map comes from New Guinea all across to the south to Tasmania. And they made a tree, and then they looked at, where the population that they sampled in Tasmania belonged to. And so it's important to know that Tasmania, the island, in southeastern Australia, there was once part of Australia connected by the land bridge, but in geological time that split, and there is a tree now, the Tasmanian Sea, and that split happened about 12,000 years ago. So the genetics showed that the split between the population of the sugar gliders from the Victorian population on the mainland, that's the closest mainland relatives of sugar gliders, that split around 150 years ago. So, this is not something that happened in an evolutionary time. So this has not split at the same time as we would expect, something like 12,000 years ago when Tasmania split from the mainland. But this was actually an introduction mediated by humans, and this actually very well overlaps with the historical data that, it's about around 150 years ago when they first show up, and then they spread across the island. So this is very important also, just highlighting that museum specimens, like all these skins stored in museums can be really important, because they repositories are very important genetic data that can be used for even today for conservation. And now that this group showed that sugar glider in Tasmania is actually an introduced species, now they can take out from legal protection, and now they can do like an eradication project on them legally. And so now I would like to talk a little bit about my current research, which also using, like all these previous techniques that I've been talking about, getting DNA, that's our main source of samples. So what we do is we work in Indonesia, and we collect the feather samples from the wild, or blood samples if we can access to any trees. And also if there's a confiscation, we also collect DNA samples from the confiscated birds as well. So in the first step, we're building a wildlife forensic genetic database, just based on the distribution of these species. We can build something that you like showed on macaws, or the Major Mitchell cockatoo, that we kind of build like a genetic map where you can see, different genetic signature on different islands. And then when there's a confiscation, we try to match all those samples against one of these populations or one of those islands, just purely based on like forensic genomics, just comparing and the two different DNA markers, and see if which population they more closely related to. And so what this can help authorities is to identify the provenance of the confiscated individuals, which is often unknown, because they confiscate a shipment, but then they have no information whatsoever where they're coming from. But we also take important swab samples from the birds to see, what diseases they also carry. So there's also going to be a biohazard evaluation of, things like avian flu. Is it out in the wild? What other diseases they might carry? That's never really been checked before on like wild cockatoo population, for example, or wild parrot population in Indonesia. And as I mentioned you, this little device that's like a small USB size sequencer, we can also develop a technique, like a test on this device, and then train the local law enforcement or local NGOs or local scientists to use and to sequence these confiscated individuals in situ, and then actually consult our database and get, a very quick evaluation. Because when there is a confiscation, there are often like, hundreds of birds. If we sequence a few birds and then know they're coming from certain islands, then the whole reintroduction project can be speeded up, and not just, ended up in a wildlife rescue center somewhere, or, even just as they often do, they just release it on the site of, or the nearest site of the confiscation. And of course, so this is like an in situ genetic analysis that we're planning to do. And the outcome of the project, what we're hoping for, and this is all an ongoing project, just funded recently by the Australian Research Council. So we just at the very beginning of the project, so I can only talk about it in theory at this point, but maybe in a year or so we can come back to this, and then I can show you some more information of what we actually found in this region. And actually we're also doing this in Mexico. So what we hope to do is to get like a whole reconstruction of the trade route and help law enforcement that way to see, where the shipments are coming from, going to, what are the main hotspots of the source of the samples. Because of course you can imagine like places like Mexico or Indonesia, they don't have so much money to monitor all these islands. But we could show, for example, as it's been done on Africa, in the ivory trade, that most of the confiscation in Southeast Asia were coming from like certain countries in Africa, not just across the whole continent. Then law enforcement can focus on specifically those regions, or we can focus on, like with education campaigns and such on those islands where the most samples are coming from, from the confiscation. So it will have law enforcement. Hopefully it can, also have, better confiscation efforts, but also rewilding. As I was mentioning, this is a live trade. If you confiscate something like an ivory or a rhino horn, unfortunately, you can put it back to the wild. But here we still have a chance, if we act in time, to rewild these birds to their original habitat. And like you all know parrots, and that they can stress very quickly in environments like very enclosed small spaces. So I think time is really critical in these circumstances to get them back to the wild as soon as possible. Of course, like following all the IUCN guidelines for like soft release and disease testing. We really hope to do like a little bit of cutting edge with this science as well, being use this small sequencer as a field-based technology. One of the first times that used for wildlife trade investigation. And, like with the disease, we can have a better evaluation of like public health implications. Of course, avian flu is a big alarm bell these days. We still remember the effects of COVID. So, monitoring is really important in the wildlife trade. And we try to do that here with the parrot trade as well. And of course, last but not least, we're going to record and document the whole project audiovisually. As you all know, like we do some videos and documentaries, and we would like to continue these efforts with this project as well, and use that for education and capacity building in Indonesia as well. So that takes me to the last part of my my talk, which is like why outreach is so important. For us academic people, scientists, publishing papers are one of the main measures of your, scientific output. It's not a perfect system, and I don't fully agree with it either. But that's what it is these days. Like you need to publish a lot of papers, your results, which is very important and fair enough. Like you need to write these down, and these can be used, later on for other scientists or for legal documents. But unfortunately, these are often behind paywall. People, the general public, often they can't access to them unless they pay a hefty subscription fee. And also these are like very much written for like other scientists, or very scientific, dry scientific languages that's often hard to understand even for other scientists from different fields. So audiovisual tools are really important. And can reach a much wider audience, not just the scientists, but like the general public, or even very specific audience, like the decision makers in the countries who, for example, going to decide to set up a program to protect the wildlife in that country. So that's why we're doing video abstracts, for example, to promote science with our organization, Wildlife Messengers, and that's one of the videos you've seen. I think that was a much easier way. If I just showed that video to you in 5 minutes, I think information comes through much clearer than reading a 10-page of a scientific paper or document. So with Wildlife Messengers, we have this slogan, film, we're making films for conservation. So, these films are specifically focused on conservation efforts and showcasing projects that work to protect wildlife. not always parrots, but that's the main theme here. You also the macabre kingdom. I think we watched it together earlier. And so our latest film also connects to Indonesia. It's called the Indonesian Parrot Project. And this shows not our current work, but our collaborators' work in Indonesia, who works, for example, on the Aboti cockatoo. That's one of the subspecies of the the yellow-crested cockatoo, sorry, the cockatoo sulfurea. And there's only found on one single island in the Java Sea. So, for example, we traveled with the team to that island and filmed this very rare population and show how they try to educate people there. So they have this conservation awareness and pride program. It's very important to to show these people and make them pride to the environment instead of selling it out, which is like a one-time income. Now they also transform people who trapped parrots earlier to protect them and to be like eco guides. And it's something that we also saw, an example in Tambopata, that it's very important if you get this thinking into people and in the future generation, they would very much do that. And I mean, they love they birds, and if they don't have to, they wouldn't want to capture them. So I think this is a great example for that as well. They have this community economic development program as well, developing supporting women's and for doing different cooperatives or different things to sell artifacts, and ecotourism as well in these local villages. For example, we went to visit one on Seram Island. That was a great experience. That's also in the film. And they also had an avian rescue and rehabilitation and release center that they revived birds. Very interesting that since the film, for example, the Indonesian government itself is supporting lots of these rescue centers, and they built some of them from government support as well. So we trying to collaborate with them with this new project of the wildlife shape. And at last, I just wanted to show you all the trailer of this film. That film is already available online on our YouTube channel, so you can watch it anytime, the whole documentary. And the last thing, if you want to know more, and of course, as I mentioned, this is just the beginning of our research, but we made a website for it. It's called stopparrottrade.org. So you can read more about the illegal parrot trade there, and see our team, read more about the project, and we tend to upload any news or communications we have, any advances in the project. You can contact us as well, and also if anyone wants to support it, it can be done through wildlife messengers or the Australian National University. We also plan to do like all these contents like videos, short videos soon through our YouTube channel as well. So if we have any reports from the field, we would like once we are out in the field and do start collecting the samples and work with the local people there, we also want to document that and keep you updated through these videos, and you will find also that all in one place on this website. And with that one, I would like to thank to all our sponsors and supporters, and thank you very much for the attention. And if you have any questions, I'm happy to answer now.
Oh, thank you very much, George. This was an excellent talk, very, very enlightening. Our club has been involved with conservation issues for many years. For those of you who don't know, the blonde woman you saw in the trailer is Bonnie Zimmerman, who has talked to our club a couple of times, including last year. If you want to see her talk, it's on our YouTube channel. And please go to Wildlife Messengers. I've seen the film. It's just gorgeous. And I would encourage you to also watch the other films there, including the Macaw Project. And George spoke to us actually a few years ago he showed the film. And if you're a club member, you've already gotten a link. If you want to get a link, I can share it with anybody who's in the meeting now. And that one is an unlisted video, so you won't see it, but it does exist on our YouTube channel. So actually, I'm going to start off because I have a few questions I've been noting down. And do you want to stop the screen share, George?
Oh yeah, sure. I think I
Good.
did.
That way we can see you there.
Mm-hmm.
So I'm just curious, when you test the eggs, well, when they tested those eggs, they obviously did it non-invasively or they would damage the eggs. Are they doing just a swab on the surface the way we do cheek swabs for genetic testing?
So the ones, for example, they found in the nest, that when there's only eggshells, of course, they can take, the whole eggshell and they can just grind it up and then digest the DNA that way. Unfortunately, when there is a confiscation like the eggs at the airport by biohazard security and confiscation, they had to fertilize those eggs. So that means that they need to destroy, unfortunately, it like very high heat, or so that would be, unfortunately not viable anymore. And then they take, I think they took samples from the tissue, actually from the egg, from the embryos, or inside the egg.
So all those eggs we saw were destroyed then.
Yeah, unfortunately, I think that's very strict. I'm not sure about Brazil how they did it, but in Australia, I'm pretty sure that's how, anything that comes through, even for us, if we don't bring samples in, like feather samples, with like with a permit, with different CITES and quarantine permit, if we don't have that on the side and they confiscate our samples, they are going to burn them all. And it's not like coming back in a week with a sample. They don't care. Like it's really strict there. Like once they confiscate something, it's gone.
Right, I understand. I mean, when I went to Australia and I read the regulations ahead of time, I saw like you couldn't even have anything that had wood in it. So I had to carefully make sure that nothing I packed had wood, because that can also be confiscated. I was just surprised the eggs came into Australia. I would have thought people would have been trying to smuggle them out.
Yeah.
I would think in that case, if they were caught at the airport before they leave Australia, then there shouldn't be a biohazard because they're Australian eggs.
Yeah,
But
so these were coming in. So they find, for example, like blue and yellow macaws and, like more that's not native to Australia, smuggling things in. Um, I'm not so sure that, that like smuggling things out from Australia is such a big thing, because of this huge biosecurity uh, um, screen at the airport. So I think it would be really hard. Although there was like a recent confiscation of eggshells. I don't know if you saw that report from ABC or BBC, and they had found lots of, I think thousands of eggs in Tasmania. But a collector who's been sending them to Europe, I think, to UK. But that was for collectors, so they were already like eggs, like just eggshells collecting. So
yeah, that was a big thing in Britain. I remember seeing the reports about that that fellow in Britain who had hundreds of thousands of eggs from around the world. And I think there was a whole documentary. I think it was called something like The Egg Collectors.
Oh, really? I didn't see
Yeah,
that.
yeah, no idea that that was going on, but it was they were depopulating birds around the world for their egg collections.
Yeah, yeah. Yeah, we also work, sorry.
I was saying it seems to be a British thing.
Yeah, I was just gonna say that I was just like last week I was in London, and we also like we have collaborators at King's College London who run a human forensic laboratory, but it also has like wildlife forensic science in it. So we work with them to try to keep all these testing at a forensic level so it can be accepted at court if has been used, and we seek they help to build this database such as the that meets the standards of like the human forensic databases. And we also had meetings with like the wildlife crime unit of the UK, and they were really interested in this whole database and using it, and they mentioning like how they could already use such information if it would be available today. And yeah, we had like meetings with the CITES headquarters as well, and they also kind of confirmed that yes, such database does not exist. There are some ivory genetic database, caribou database, but nothing yet on parrots, and you're kind of surprised because it's such a big trade on parrots. So we really hope to have this database useful.
Yeah, in fact, I was just reading today that Bangladesh was just removed from CITES because of uncontrolled illegal parrot act. Well, I shouldn't even say illegal under CITES, but apparently I didn't know Bangladesh is a hotspot for the parrot trade, and they show no inclination to stop it. So CITES shut them down, though CITES is a voluntary organization anyhow, so I'm not really clear what that accomplishes to just remove them from something they weren't following anyhow. But
yeah.
And let me see, Kim wrote, well, first of all, thank you for all the information. And then how do airport employees discover smugglers like the egg vest?
Yeah, it's a good question because it's not going to show up on the detectors when you go through the metal detectors. So I guess it's just often it's just a luck that they find, for example, that person with the vest. Maybe there was something suspicious, and then they like sometimes they just like randomly screen you and touch, and then they can probably feel that it was not like a six-pack on the radar one, something.
Maybe
Uh,
it looked bumpy, and maybe it wasn't hidden
yeah,
daughter.
yeah, and yeah. You wonder like how many other people might just sneak through um, with things like that. But and this is Australia with very high alert on that. You can imagine like other countries that don't
Well,
have such.
it's funny, studies have shown that people tend to touch the things they're concerned about. So I've read that one of the way pickpockets operate is they sort of brush up against you, or let's say they brush up against a man and they want to know where his wallet is, and you might not be able to tell, but they brush up against him, and his first instinct is to touch where the wallet is to make sure it's okay. And meanwhile, they haven't touched the wallet yet, but now they know where the wallet is and they can take it. So I want people, it's hard for people to not look guilty when they're committing crimes. So perhaps the egg smugglers, when they're going through security, they might be like touching their chest repeatedly to make sure the eggs are still there, make sure that they don't look visible.
Yeah.
Themselves a lot on the chest, that probably alerts security people to go, wait a minute, why is that guy touching his chest a lot? Is he smuggling something underneath? They may not know it's eggs, obviously, but there may be,
Yeah, of course,
this hot belly and he keeps touching and that looks suspicious. So we should pat him down.
Yeah, and I'm sure they're not looking for eggs in those instances. They're looking for drugs or something. And there is a really good series. I think it's called Australian Border Protection, Border Force or something, Australia, which is great. Like if you watch and you see, those are like, some of them are real things, and just the things that people can smuggle different things. It's just yeah, really interesting. And of course, some people who are not even aware of these, what you can bring in and out, and then often it's also just like not intentional but unintentional. Yeah, so
yeah, interesting. So I want to ask you, in the field, you've got that rapid sequencer. First of all, what do you use the specimens? Because here, let's say we want to sex a parrot, we pluck a feather from the chest that has a little blood at the end. So what do you do with the wild birds? And then how long does it take you to actually do the sequence in the field?
yeah, so it's so it depends on what we use for like setting up like the database and to get like a really high quality like reference genome for a certain species or a subspecies. You really need like a lot of DNA and really high quality DNA. So that high quality DNA can be found in bird blood, because they red blood cells, they still have a nucleus, not like mammals. So there's like a huge content of DNA there. So we often use like blood samples for setting up the database and such. But I think that would be a bit complicated. We were also talking about this with the police in the UK, that would be a bit hard for like just someone like a border protection officer to take blood, of course, without a vet. So once this is all set up, and for just the regular screening and the field or people, it's much easier, as you said, just pluck a feather. It doesn't even need to have blood, just some fresh tissue. So the first step is you need to extract that DNA out of the feather. And then there's also some quick procedures, like also these CSI kits that can do the DNA extraction within a few hours. And then you have a small liquid that you have the DNA in it. And then use that liquid to pipette it on this little device. And then, it's a bit, like you need to add other liquids, etc. But it's quite a protocol you just need to follow. And then you run the sequencer, you connect it to a computer like your laptop, and then you run the sequencer. The length of like the time, how long you need to or you want to run it, depends of what you're interested in. So, for example, if we only want to do just like a few genes on the mitochondrial DNA, which are quite short, you can just sequence and run it, for example, up for an hour, and then you already have that information, or even less time. And then you can see on your screen actually how these, things are reading the DNA out. So it's really amazing. It's like live sequencing on your screen. So you can have this information fairly quick, definitely the same day of the sample collection, which is amazing. Like thinking back of the first human genome took like, how many decades to sequence, and millions and billions of dollars. and like one sequencer like this costs something like $1,000 with the kit included. So it's not that much, and it's going down, further down. But if you're interested, like something like a whole genome, that's also possible, but it takes a longer time to sequence. But what we would like to do is really focus on certain parts of the genome or certain markers that we always use the same markers. So then we have this setup that we just need to sequence some part of the DNA, so that it doesn't take that much time.
And you'll be reliably able to distinguish between the different Indonesian subpopulations of cockatoos.
Well, that's what we will see, but that's what we expect. Yeah, that's what we hope. And that's why we need to collect these samples from the wild first. So we first understand, what the diversity is, what the genetic structure there is, what's the allele frequency, like what's the genetic diversity between these populations. If there's not much diversity in a species, for example, a different region, it's really hard to find and differentiate the populations. But usually, like if you think, for example, the eclectus parrots, you have these very I think like 10 or something like 9 subspecies in the wild. So that's a good reflection that they also have a big genetic diversity in the wild that will be indicative of their provenance.
Yeah, I was surprised when I learned that about how many subspecies of eclecti they are. And they do have some feather differences, but I don't know how
Yeah.
that can be used, particularly if there were to be, for example, interbreeding. But
Yeah, exactly.
now, when you do the cockatoo genomes in the wild to establish the database, do you do fuller genomes? Or do you know where the markers are already that will distinguish between the subspecies? Or at this point, are you trying to find out what markers there will be to distinguish?
Yeah, so for the in general, we have it's just recently this year it came out the new phylogeny of parrots. It's been a big work on and they try to include it's a collaboration with American Museum of Natural History, with CSI in Australia, and they included almost one species of parrots of all these 400 to have some kind of genetic resolution in it, and quite a big one. Well, we try to do even higher resolution and look into even within the species diversity. For that, unfortunately, we don't have yet samples and genetic information across all the species. Talking about the eclectus, for example, now it's recognized as the 4 different species and 9 different subspecies, but it's still a little bit iffy, like the whole diversity. And as you said, for example, the females, they have some differences, patterns, but of course you need to have them, side by side and comparing. But the males, they kind of just green, it's really hard to tell them apart, even with an expert. Imagine someone just confiscating them, as a police would be not much clue about it. So I think it's much more reliable once we have the DNA, and it's something that's more that you can measure, and you don't need to bring always an ornithologist or a parrot expert to identify the birds for you. But yes, exactly. That's the first step to get out, to collect samples or get from museum specimens, and then sequence the whole genome. And then once we have the whole genomes, then we can align these genomes, and then we can screen and look at differences. Like this could be single nucleotide polymorphism, just one single site, or that could be these different repetitive regions that human forensics is still using traditional, and that's what we use for macaques as well. So these are different genetic markers that we can look once we have the whole genome sequence. And so that's part of the project, as you said, yes, that to develop these markers first, and then in the future, then we can only look into that specific region of the genome to find them.
This sounds quite expensive. I mean, if it's going to cost you $1,000 a sample just to create the database and then to test, are you relying on grants or ANU support, or this sounds like a very expensive project.
Yeah. Yeah, so that $1,000 is actually not for one sample, like because that's like you can fit multiple samples into that. So we kind of tag these samples with a specific barcode. And then once we have the data, we know, like, on a computer, we can match which individual or which sample this belongs to. So you can make hundreds of samples on the same sequencing run. So you don't need to do them one by one, which is also a great for cost. So it actually, for cost, it can drop to, like $10 or $20 per sample, and we tend to push it even further down, so it would be something feasible, because otherwise it would be so expensive that no one would really do that. So it's also important to have, like, in situ and cheap and transferable to other labs, so everyone can do it easily. It's not, doesn't require now not an expert but a geneticist to run it. But yes, at the beginning there are some investment in cost to set up the database, as I said, to generate those whole genomes reliably, and also at the forensic standard, like, that we have these genomes. We know that these individuals are coming from that location because we collected them, and we know that they from there. And for example, we couldn't use like a pet macaw to sequence the whole genome for this case, because that could be at some point hybridized with another bird, or we don't know the origin of that bird. So that's why we really need to rely on wild samples or museum specimens with location tags. So yeah, we have like grants from the Australian Research Council that supports, and that's one of our main funders. And we also got from the Loro Parque another support for like the field work. But as you said, yes, the genetic part is also like a big complement. But we have, like, yeah, other institutions as well as like putting in into the research. So yeah, fingers crossed that we can get. And also, this is like, we're not trying to we hope to resolve this big issue, but we can't really focus on all the parrot species traded at this point. So this would be just the first kind of a proof of concept to build up database on a few species that are heavily traded or most traded, and then kind of show that what it can what it can achieve, how it can help. And we hope that once this has been demonstrated and published, then we can get some more funding to do more populations, more birds. And this is something that we can just, it's not a one-off, it's a database that can be expanded later on.
Wow, this is tremendous. And I mean, I'm always hearing about them extracting ancient DNA, so I guess you can still get pretty good DNA from the museum samples as long as they're accurately labeled. So that's the thing you have to be careful of. If they say this cockatoo came from such and such island, but they didn't, that could be misleading.
Yes, exactly. And you just mentioned ancient DNA. I was last week, as I mentioned, I was in Spain. Actually, I was visiting a friend and colleague of mine in Spain who just established an ancient DNA lab. And that's very similar to the protocol. Like, you need to be all masked and all suited up, because the contamination in ancient DNA is actually, it's really, it's a big issue. And especially when working with humans, and us being can contaminate the samples. We just had we have another project on looking at ancient macaw DNA that found in a tomb in Peru. And we actually successfully extracted ancient DNA from those feathers of 600 or something years old. And that was a big achievement, to have such a minute amount of DNA extracted, so avoiding contamination. But then forensic science is also very, very careful about, like, not contaminating your samples, because like, these samples that coming from the wild, they have to be in such a high standard that you because it's going to be used as like a standard for other comparing to other samples confiscated. So yeah, you need to be very careful with like yeah, ancient DNA labs or forensic labs. They're all very yeah, precise ones.
Wow, fascinating. Now, does anybody have any other questions? I feel like I've been hogging this a bit. You can put it in the chat or unmute yourself if you want to ask any questions. Oh, I see one unmute.
Hi. Do you think there are any ramifications for current parrot owners from all of this?
Well, the one we were also talking about with, law enforcement is that, knowing that there is also like a legal side of the trade, and that actually help to alleviate, the most of the illegal poaching from the wild. Certain breeders, like they produce lots of they breed lots of these parrots in the breeding facility. But then when it comes to the legality or illegality of certain birds fed into this system, it's also important to have some reference from these breeders, these reliable breeders. So what we were thinking is to try to collaborate and work with breeders who can, like send in, or we can go out and take samples from the breeding pairs. And then that also includes to the database, and that can show, like if there's like a confiscation or a check or some doubt that this bird is legal or illegal, then we can show that, oh, this is actually belongs to these breeding pairs. So that's actually okay. That's a legally traded bird, and that comes from the breeder. But compared to if it matches with one of the wild populations and not like one of the breeding facilities, that's an alarm bell that this might came from the wild. So I kind of see that kind of assurance, for keepers, for parrot keepers who want to buy parrot in a responsible way, that hopefully this database can also, give you some kind of certainty, because even CITES papers can be falsified these days, and it's very often the case in many places. Actually, in Indonesia, we had like cases that they actually show that they imported white-coat birds and made them banded them and then gave them CITES papers. So, for example, one of my students in Indonesia, he's looking, or he's going to look for his PhD, different isotope patterns of the feathers, birds that are sampled in the wild and birds that are kept in like captivity or in a breeding facility, because like you can tell them, what seed or what food they were raised on, that builds into like an isotope pattern of the feather. So, for example, if you just take a feather sample from that cockatoo that we mentioned earlier, that this person just poached from the wild, then you could show that this bird was not raised on some kind of pellet that they like they raise, but this is like raised on a natural food, so it's coming from the wild. That's even like a more direct and quicker identification of like separating like traded or hand raised birds. So
Wow, that's amazing that they could do that.
That's
yeah.
awesome.
Yeah, it's often used for like human, like anthropologists as well. And when they excavate bones, and sometimes if they, it's also cheaper actually, to get an isotope test than a DNA test or an ancient DNA test in that case. And they can see, that those people who lived in that area, if they were from that region, just based on the isotope pattern of, the rocks, the minerals in that region, that they would share. If it's the same in their bones, that means that they've been raised there. But if there's like an outlier, then you can see like this person was, for example, a migrant from a different location, and often they can even identify those locations, similar to like genetics we do. There's also like different isotope landscape, isotope patterns in different regions, based on different mineral distributions, and that also reflects like a parrot in the Amazon who feeds on certain fruits. Their isotope pattern is going to be very different from from a bird that eats seeds in captivity. So that's another way. But yeah, going back to your question, I think this could be some kind of like certification system as well, to to find kind of like the human like missing cases, when you cannot really identify the person who's been missing, but you sample some of their relatives, and you have the relatives in the database, then you can have like a close match. And that's could work like that as well. If we know what individuals are in the legal trade among the breeders, then we can separate them from the wild.
It's amazing work, and I hope that law enforcement will do something with it, because it's one thing to make all these identifications, but if you have places like Indonesia or Bangladesh that aren't going to enforce any of the rules, and it doesn't really matter what data you have.
Yes, exactly. That's a big question. And as I also mentioned, many of these countries, it's often, even if you have the evidence, it's not that important. But that's why, we also try to do with all the outreach campaign to show, why this is important. And then hopefully, even local government might react. They actually are very, very proactive on confiscating birds in Indonesia in the last years. And I'm sure Bonnie talked about that as well, that we actually have more confiscation in Indonesia than the places they can, like hosted in rescue centers. So I think there is a big movement from the government as well, trying to be more alert, and I really hope that this could be also, like an extra tool, something that they can channel their funding in a better way in the future.
Yeah, education is so important.
Yeah, very
Well,
much
I mean,
so.
you're doing amazing work on behalf of Parrots, and I've always admired the work you do. I can't wait to hear, 2 or 3 years time how the project is doing. So
Yeah.
I want to have you come today to tell us about the project, and then we can see in a few years, how that's working. And
Yes, definitely. That would be so exciting, wouldn't it? Just showing like what we found and what the evidence shows, and yeah, make a recap.
yeah, be fantastic. Does anybody have any last questions? Again, you can unmute or put them in the chat. All right, well, then I want to thank everyone for coming today, and it will be posted to YouTube. I'll be sending that out, or you can take a look on our channel, and I'll send that out to everybody so you can watch it after the fact. And George, thank you so much for taking the time to talk to us about this. This is such exciting research. I couldn't wait to hear more about this. So
Fantastic.
So that's for the opportunity.
yeah, it's really, really great stuff. So thank everyone for coming. Hope to see you again, and we will end the recording now.
Thank you.